Okay, so we've talked about the nervous system as far as the central nervous system, right? Already last week we dissected brains. Yeah, you missed the brain dissection. It's pretty cool. It was actually fairly unremarkable, right? It's like you're looking at it going all look kind of the same, except for that diencephalon and
it just looked like all white matter.
You think even on the cerebrum?
Well, the outside, but like the inside just looked like. Yeah, you would think there'd be like stuff going on inside, but it's.
I know.
Just like,
and to think of how much stuff it does. Of course,
right.
It was a lamb, and lambs aren't very smart.
Like how does
so.
like stuff that looks like just like meat like do all that?
I know.
Crazy.
Does all that crazy stuff that's the best computer ever invented, right? So, all right, so we're going to move kind of away from the brain and the spinal cord and toward the peripheral. Remember, peripheral out to the side. So this is going to be the peripheral nervous system. Do y'all remember I made you a chart that had this kind of flow chart? Do y'all have that by any chance?
No, but
I forgot my entire bag, so I don't have it even. So, but it was like a little chart that divided out what the peripheral nervous system does. It would be helpful to study for the quiz next time if you had that. I just had printed it out for you.
I have it.
And we're going to go through it. I'll have a little bit on the presentation here.
Is it the division of what bodily functions?
It said I think it was all peripheral nervous system, right? That one.
Okay.
Yeah, this one right here. Yeah, the one that looks like this.
I have it.
Okay. So yeah, have that in front of you. You can take a look and it will help you with this. Yeah. Okay. All right. So the PNS stands for peripheral nervous system. It's going to be talking about the nerves. And the nerves sometimes all get together in certain places, and we call that ganglion or ganglia. And we will not talk a bunch about that, except we'll see places where the ganglia are, namely in some nerve junction areas. Okay, so on your chart you have the two divisions. We have our central nervous system here, which is the brain and spinal cord. And then we have our peripheral nervous system that breaks into the divisions of afferent and efferent. And remember, afferent, I wrote on your chart, is a feeling. So we're going to be talking about senses today. All right, and then our efferent is going to be our motor. So afferent, a feeling, efferent, email. Right, it's going to send the email back to the brain. We have our somatic, and then we have our autonomic. Somatic would be voluntary. Autonomic would be then involuntary. And then that's broken down into two further groups, the sympathetic and parasympathetic. Does that look like it's following that chart there? Okay, and I even have a little more information on that chart as far as first thing short, the other second one, second axon long, that kind of thing. Okay, and we're going to get to each one of those things. So I would recommend memorizing that chart. You may not memorize it, but look at it, study it. It'll be helpful for your quiz next week. And next week we're going to be talking about our senses. In fact, it's going to be we have to cram everything in here to talk about all the different senses. Okay, here we go. So the efferent neurons send signals from the CNS to the effectors, so the email, right? It's going to send it from the CNS, the central nervous system, to the effectors, all right? So I don't know where things are going the wrong way here. Here we go, efferent, right here, efferent on the bottom. From the brain, this is showing the spinal cord. So remember, spinal cord is like a little tiny miniature brain that's running through your back there. The gray matter right in there. We're going to see ganglion, a lot of ganglion in there. It's going to send it to the muscle and tell the muscle to move. So efferent, email, muscle moves. Afferent, a feeling. Okay, up on the top here, from the skin to the spinal cord to the brain, I just felt something on my skin. It itches. I got stung by something like that, and that is being sent to the CNS. All right, so afferent, efferent, yes? A feeling, email. All right, the efferent PNS, the motor division. Okay, the somatic motor nervous system, SMNS, is that on your chart? SMNS, somatic motor nervous system. It controls the voluntary muscles. There is one neuron and it's myelinated. Okay, so one neuron going from the source, brain, spinal cord, to the effector, the muscle in this case. It's going to be one neuron that's myelinated. There's only one. Can y'all see that? You can see it okay? It looks a little dim to me. Maybe I should go in there and bold it or something.
I see it fine, it's just the text kind of thin, so it's kind of.
Yeah, I need to make it bold, I
A
think.
little higher in the eyes.
If I turn
seems like
the.
there should be a place for us to write this, and I don't.
it better here, but can you
Yeah.
see your notes?
Yes.
Okay. All right. It has only one neurotransmitter, acetylcholine. We know about acetylcholine. That's on the chart I made for you. Okay? All right. And the last thing is it's excitable muscle tissue receives a signal and it reacts to that signal. The muscle will move. Okay? So that's what's happening in the efferent PNS motor division, the SMNS. Somatic motor nervous system. The efferent is the autonomic, or automatic if you will, controls the involuntary muscles. There are two neurons. The first is myelinated, the second is unmyelinated. Is that in your book?
Yeah.
And y'all can see okay?
Yes.
All right, neurons can be excitatory or inhibitory. We actually know a little bit about that from our muscle unit when we talked about sometimes our neurons touching the muscles or keeping it from contracting, right? They're stopping the action. There are two neurotransmitters. This is new information. We have ACH, acetylcholine, and something called norepinephrine. Norepinephrine. Sometimes I just call it nor to shorten it a little bit. This is on the chart that I made for you. See how helpful your chart's going to be? Two further divisions are the sympathetic and the parasympathetic. It's going to be divided down. Remember, this is all autonomic. These are the things we don't have really any control over. It's involuntary. The brain is handling it for us every single day and night. Do y'all have some just blanks to fill in? Is that what you're doing?
Yeah.
I can't even look at my book because I forgot my book, my bag. And you can refer to that chart I gave you as well. See, you can see it okay? It actually looks better from back here. Okay, I'm gonna have to move on, y'all. So here we have our autonomic motor neuron right here. Take a look. It's coming from our spinal cord / brain, coming to it right here. We have the first neuron and then the second neuron, right? Right there. And there's some place here, a ganglion, where they're having a connection. Remember, ganglion's gonna be kind of that bundle there. All right, the first one is myelinated, the second one is unmyelinated. Interesting. I don't know why. We can either use acetylcholine or norepinephrine to touch the effectors in some way. Okay? We call this the preganglionic neuron and the postganglionic neuron. What's the ganglia right here? Before it and after it. That's all that means. And you'll see a picture that looks a lot like this in your book. All right, sympathetic versus parasympathetic. Now we're going to go to that breakdown and go through those. The sympathetic division covers the whole body. It takes over in times of stress. And the first neuron is short and the second neuron is long. That's just a difference between the two. Our parasympathetic division covers the head and trunk only. It takes over in times of rest and calm. The first neuron is long and the second neuron is short. That's just information of the anatomy of it. I don't know that there's anything more significant about it. And that is, by the way, also written on the chart. There's a lot of stuff I wrote on the chart for y'all there. So it's right there for you to study it that way. All right, so we're taking now our sympathetic, which takes over in times of stress, and our parasympathetic in times of rest. And remember, these are both in the autonomic division. All right, so this is all involuntary. So you're going to be kind of maybe surprised about how much involuntary stuff is going on in your body right this minute. Okay, you don't have to think about it at all. Your brain is so good to you that it's taking care of all this stuff. All right, so we're going to take a look at some of that. Are we good here? Yeah. All right, what are we going to take? Oh, here we go. In times of rest, our parasympathetic division versus our sympathetic in times of stress. All right, so what happens during times of rest? Right, we're going to see several things here. Our heartbeat can be slowed down as we're resting, just recuperating from some stressful event. Our heartbeat will increase if we're in times of stress. Look at the eyes. The pupil will dilate and get really small when we're stressed out. It will get bigger when we're resting. Saliva flow. This is interesting. We have more saliva flow during a rest time than we do during stress time. If you think about if you've ever taken like an English class where you had to give a speech and stand in front of the class or whatever, and you're a little bit nervous, kind of feeling a little bit stressed, right? And all of a sudden your mouth feels really dry. You're like, oh, I need to piece of gum or something to get my mouth. That's because the sympathetic division is in charge of your body at that time. You may have a heartbeat kind of racing a little bit. We're going to see a dilation of the bronchi in the lungs. And it means they're getting bigger. And if they're getting bigger, that means I'll have more oxygen flow. All right, so I can breathe and get more oxygen into my blood to accommodate for my accelerated heartbeat. Constricted here to make it, I don't need it as much oxygen at that time. We're going to see, peristalsis get stimulated. That means I can digest my food during times of rest. That's interesting. We're going to digest during times of rest, whereas not so much during times of stress. Release of bile, that's helping in digestion. Contracting of bladder, meaning I can empty my bladder. We need to be at rest in order to empty our bladder. If we're too stressed out, your bladder's going to be like, I'm not going to do it. All right, you have to calm down to urinate. All right, so many functions are controlled by either the parasympathetic or the sympathetic. One or the other only takes care of it. But there are other things. Okay, for instance, the lacrimal glands are completely in the parasympathetic. Lacrimal glands are the glands that produce tears in your eyes. Sympathetic covers the whole body, so the dilation of blood vessel and limbs and the erector pili, those are the muscles that make your hair stand up, right? Yes.
So lacrimal glands, the ones that make you cry, they're when you're at rest?
Yeah, apparently our lacrimal glands are, does it tell us up here? It does not tell us.
Why would you cry when you're at rest?
Well, what it means is that you have, it seems like when you're stressed out. The problem is when you're stressed out, everything kind of, I must say, dries up, for a lack of a better term, right there, you know. So, but the parasympathetic's in control of it. Okay, that doesn't mean that just because you're stressed, the parasympathetic has nothing to do. It can still manage there. But it does tell us the parasympathetic right there. Redundancy in the sympathetic. The stimulation of adrenal gland will increase heart rate, and it's also directly associated with it right here. So redundancy means there's more than one way to get there. All right, so getting your heart rate to the place it needs to be is pretty important, and so therefore we have a redundant system to get your heart rate up to accommodate the muscles or whatever needs the blood at the time. All right, so this you're going to write down in your little chart thing. All right, do you need me to turn the light on for this? You can still see it, yeah? The autonomic nervous system controls, this is a summary of the autonomic, heart rate and blood pressure, digestive and salivary glands. So heart rate, right, it's both because it's going to be increased during sympathetic and decreased during parasympathetic. That's going to be the same for blood pressure right there. Digestive is going to be increased during parasympathetic and decreased during sympathetic. Urination, skin contraction, respiration, vision, and lacrimal glands. Liver and glucose levels, adrenal glands, and hormones. So all of those are being controlled by your autonomic or your involuntary system. How are we doing? Yeah, so I looked this up. It says parasympathetic neurons stimulate the secretion of lacrimal fluid, tears. But it says that there's also some control in the sympathetic. There's some innervation in the sympathetic. So apparently it's complicated. The nervous system complicated. All right, I gotta turn the light off for this. Okay, so check this out. We're gonna start here in the middle. All right, we're gonna see what happens. This is the control of the autonomic nervous system in general. So look here to the left, we're gonna see blood pressure increases. So for some reason my blood pressure is going up and my body's gonna react. Remember, the body's always trying to achieve homeostasis. It's always trying to achieve balance. So it's gonna try to counteract that by going through a process. So what happens is a receptor on the arteries of your heart, namely the aorta, are going to detect the increase in blood pressure. The receptor is gonna send a signal to the associated neurons in the medulla oblongata of the brainstem. The association neurons, that's neuron to neuron, send a signal to the parasympathetic neurons. Parasympathetic neurons then send a signal to the heart, causing the blood pressure to decrease. Okay, so the parasympathetic's in charge of trying to get us out of stress and back into rest, okay? We don't want to be stressed. Our body doesn't want to be stressed. We want to be in a, right, calm time. Now, we're going to start over here on the left side again. Blood pressure decreases for some reason. It drops for some reason. Some people may not have eaten the right amount of food at the right time. Blood loss can also cause a sudden decrease of blood pressure. All right, so a receptor artery detects the decrease, aorta. The receptor sends a signal to the association neurons in the medulla, same pathway. The association neurons send a signal to the sympathetic neurons, and the sympathetic neurons send a signal to bring the heart rate up. Okay? So you see how they're working together. You can even say counteracting each other, all right? Trying to get everything into a place of homeostasis. All right, here's the fun part of Module 9, sensory organs. We're talking about the afferent division right now. That is the fun part of Module 9. Oops, sorry. An organ which responds to a specific type of stimulus and triggers an action potential, which is a signal, on a sensory neuron. A sensory neuron. Remember, sensory means has to do with the senses. Can y'all name the 6 senses for me?
Touch.
Vision, touch, taste, smell, hearing,
Sensory.
balance.
Oh,
Balance, they kind of added that later because it is achieved in a sensory organ, which is your ear. Okay, we'll get to all of that. All right, here they only put the classic ones on there. Vision, hearing, smell, taste, touch, and we could break that down to also balance right there. Okay, the afferent PNS, A feeling, the senses, and the receptors, there are 3 classes. You're going to write these down. Can y'all see well enough? Okay. Somatic, remember somatic means body, right? So soma means body. That's touch. We're going to see the skin, the muscle, and tendons are going to have receptors for touch. Okay, if you've ever had a deep wound, you know that there are some deep sensory organs there that can, we call that a visceral. Here we go, right here. The visceral one is going to be deep. So near the surface, we have a somatic receptor. Deep down, we have a visceral receptor. I don't know if you've ever known anybody who had a kidney stone. It's painful. Very, very painful. It's deep in the body. Then we have special receptors, or specific sensory receptors that are found in ear, eyes, tongue, and nose for those special senses. Okay, we're going to see that these are special senses with special, highly individualized receptors, and these are going to not be special. The sense of touch gets left out a little bit. And so what we have to do today, y'all, is to go through the general deal about the receptors and each thing, and then specifically about each organ, okay? So I think we're going to have to, like, I feel like I remember this lecture taking, like, the whole time to get through it. All right, so receptor types. There are 5 types. 1, mechanoreceptors. Mechanoreceptors. I'll give you a hint. Receptor is going to be at the end of each one of these. Mechano means movement or mechanism, right? Or machine. That's responding to motion, and those are found in the skin and in the ear. The ear sound that you're hearing right now in your ear is because of the motion of the air that my voice is traveling to you. Okay, it has to travel, sound has to travel through something. And right here you're hearing my voice because my voice is traveling through the air. And it's moving. It's moving in your ear. We'll see that specifically. Next, thermoreceptors. Thermo, heat, right? That's responding to temperature. So the sense of touch. You can feel something and know that something is hot or cold. So any kind of temperature or temperature difference. Number 3, photoreceptors. They respond to light stimuli, and those are found strictly in the eye. Okay, the eye has photoreceptors to accept stimuli for vision. Number 4, chemoreceptors. They're going to respond to chemicals. That's going to be the sense of taste and the sense of smell. Those things will dissolve either on your tongue or up in your olfactory epithelium, and you'll be able to, depending on the chemical, be able to smell it or taste it. And then the last one is called a nociceptor, noci, and that has to do with overstimulation and pain, like deep pain. So the nociceptors will be used when you have kind of a serious injury or something like that. Sometimes they'll take over during times of overstimulation. I don't know if y'all ever did this, if you know about this. We used to take the babies for a ride in the car if they got real fussy, because all the noise and motion in the car would overstimulate them and they would just fall asleep. I don't know if that's considered abuse or anything, but my grandkids don't get soothed by the car, so it doesn't work for them. So it turns out that these receptors respond to these certain things in certain places. All right, so if there is a stimuli or stimulus that is to one of these and it's improper, then there'll be a way that the organ will react in the only way it knows how. Okay? So I'll show you an example of that once we're done here. For instance, if you get knocked in the side of the head around where your ear is, and your ear gets it's hit, like maybe in a football game or something, the only way your ear can respond to that is to make a sound. So you might have a ringing in your ears. Okay, sometimes that can happen even for a loud noise coming into your ear. Yes?
Is it normal to have that ringing noise consistently?
No. That's called tinnitus or tinnitus. It's very, very annoying. Do you know somebody who has that?
I have it.
I constantly hear ringing.
All the time? Okay, that's called tinnitus, or
I've gotten
sometimes I
used
pronounce
to
it
it.
tinnitus. My husband has that too. Yeah, and so it should be, there's something in there. I don't think we completely figured it out yet, what causes tinnitus.
What about people that can like turn it on and off?
Well, so I know there's a therapy that they use that you can practice and but it takes a lot of work on your part to get rid of the tinnitus. So my husband has that, and it bugs him a lot, but he's not willing to go figure out the practice he has to do to get it.
Because my
he won't
mom
know.
and I both, it's like a muscle or something that you can move, and you can make like a buzzing noise in your ears, and then you can stop.
Oh, I can do that.
Oh, really? Okay, so that probably is part of what the therapy is. I don't know. I can't.
Yeah, you like-
Yeah, I don't know. I
Yeah,
can't.
right.
Yeah.
I don't know how to do that. Yeah.
I know that.
So if you have tinnitus, maybe you can manipulate that to stop the ringing. Ringing in your ears when there's no noise
I thought that was
is
normal.
annoying,
I thought it
right?
was normal,
Mateo,
yeah.
is that annoying?
I've gotten used to
You've
it.
gotten used to it? Is it both ears or just one?
I think it's both.
Okay, I have it happen in one ear now and then. You know, I'll say, oh, there it is, and then it'll go away for
I
me.
usually
Oh,
only
yeah, that's
don't
fine.
hear it when
Yeah.
I'm completely not focused on anything.
Like, do you hear the fuzzy sound like when you're yawning? Like when you yawn?
I've heard that
fuzzy
before.
sound,
That's like that little.
You can turn it on and off. That's
Okay,
it.
all right. The other more classic one, of course, if you get hit in the back of the head, boom, in your occipital lobe, right there. The only way your occipital lobe has to deal with that stimulus is in your eyes, okay, because that's the vision part of your brain. And so that's the seeing stars thing. Have y'all ever been in a position where you saw stars?
Yes. That's
Yikes, no good. You may have had a concussion if you saw stars.
I've heard that if somebody gets hit hard enough on that little bump in the back, they can kill them.
Cool. Yeah.
Well,
Is that true?
there are head injuries that can take place that can kill you for sure, but it's not that common, right? So otherwise, but that's why the guys in the football field wear the helmets. That's why they wear those to protect that part of their brain.
Is
stuff like
this
that.
the only spot that'll make your eyes do that?
Because it's the occipital lobe.
Well, over the weekend, I probably have a broken nose right now because
Uh-oh.
I was goofing off and I slipped and fell, and our piano bench was kind enough to catch me, and it hit me right here, and that's why it's all messed up. And this whole eye went
Well, that's probably because it hit like a nerve that is to your eye. The big nerve comes in the back, but there are nerves all the way around it, muscular nerves really, and those are strictly for the sense of seeing. You know what I'm saying? They're not, although it may hurt. You could have had some pain.
What if your eyes just do that regularly?
Like a flash,
No, like you just start seeing random things.
random things like
no, like
lights?
schizophrenia, but like
but like
things
people,
will start moving.
like
on
things
their own.
coming across your, like the floater thing going
Yeah,
across your
yeah.
eye. Yeah, that's pretty normal. Those are just cells that are moving through the fluid of your eye. Means you have a lot of fluid.
It also happens when you're dehydrated, right? Like if you're
It can.
dehydrated and you stand up too fast, you're like,
Oh, that happens
Uh-huh.
to me
Oh,
all the
yeah.
time.
Yeah.
Like when you get lightheaded and
I
it
don't
goes
even
dark.
know.
Yeah, that a lot of times has to do with the dehydration in your whole brain, not just your eyes. Your eyes just, that's the way that part of your brain is dealing with it, okay? Yeah. So yeah, but we call that an appropriate or inappropriate stimulus, and the only way we can react to it is the way our brain in that position reacts to it. So if somebody, for instance, had a brain tumor, which sadly we don't want that to happen to anybody, right? It could be in a spot that would stimulate that, you see, so that might be where they're seeing something. I know that as far as a flash of light, there's a type of a migraine called a ocular migraine. Doesn't hurt, but it gives a flash. And I think I've had those recently, and it doesn't hurt, but the next day I feel dizzy. And it has something to do with that. So
So is that where schizophrenia is? There's something wrong in the vision part of their brain that makes them see stuff?
no, I mean, I'm not saying it can. I'm saying when you're talking
Talking
about
about
that,
our brains
schizophrenia is
being
something that has to do with the
manipulated.
chemical balance in the brain. So there's, I mean, I know we have medications to balance those chemicals out now, versus remember the frontal lobe, yeah, that thing, we don't wanna do that, right? We don't wanna go messing with the brain, to do that, yeah. Alright, our receptors can also be classified as simple or complex. Those are blanks. Do they have to fill those in? Simple or complex. I will tell you, simple is an oversimplification because nothing's really simple. Everything's pretty complex, but because of the way it works, we look at it as a simple. Simple is found in the skin, and complex is everywhere else. In other words, complex has to do with the type of receptors. Remember, there are special receptors. Those are found here, eye, ear, tongue, and nose, and skin not, right? So our general senses versus our special senses. The special senses are the eyes for vision, the ears for hearing, the ears for balance, smell is the olfaction, that's the nose, you could say, and the tongue for taste. Okay, those have special receptors, and therefore they're known as the special senses. Therefore, the sense of touch is considered simple or general, even though it's not simple. So this, you really pretty much already know this, right? Anything new there to y'all? Did y'all know olfaction meant the sense of smell? The olfactory bulb. We talked about the olfactory bulb in the brain. And you can see over here our special senses are all up here. We see our tongue, our ear for hearing, ear for balance. This is the sense of smell, both these pictures here, and the sense of vision, these pictures down here, the eyeball. We're going to get to each one of these separately. Next week we'll dissect an eyeball, and we'll do some other cool stuff, actually,
Like cow
about our
eyeball?
senses. It might be a sheep. Sheep or cow. Cows have bigger eyeballs, for sure. Those are pretty fun. Eyeballs are fairly impressive dissection. If you took general science with me, did we do an eyeball? Seems like we did.
Oh, yeah.
The eyeball and the mouth.
Yes, I remember taking that home.
That was gross.
Take me home.
No, we took it home
An eyeball?
because it was during COVID.
It was
Oh.
COVID, yeah, COVID spring. Yeah, I had to send a bunch of stuff home. Yeah. All right, and so we're going to be looking at our general sense is going to be the sense of touch. So we're going to start there with why is touch not special? It just has to do with the receptors being more general. We have thermoreceptors, we have nociceptors, we have mechanoreceptors, different kinds for different things. So touch is mediated by somatic receptors. Remember, somatic just means body. Two types. We have cutaneous. Those are going to be higher up. Those are in the skin. They're going to be closer to the surface of your body, basically. And the proprioceptors are in muscles and tendons. Those are more visceral. They're going to be deep. Okay, and we're going to look at each one of these little things right here and talk about the different types of receptors and what they do, how they sense touch. Because touch is actually more complex, really, then, and then like vision, you know, you got light coming in your eye, boom, there it is. With touch, we can be feeling heat, we can be feeling cold, we can be feeling light touch, we can be feeling deep touch, a pressure touch, a pain touch, all these different things. And all these different things here respond to those. All right, so this slide you have cutaneous and proprioceptors. That's what you're writing down on this slide. So it may not be special, but it's general or simple. All right, so simple somatic. Here we go. The simple sense of touch is located in skin, muscles, and tendons. And we're going to see here, oh goodness, can y'all read those? The cutaneous ones include free nerve endings, hair follicle receptors, something called a Pacinian corpuscle. That's cool. Ruffini's organ. That is for like a stretching. When you, has anybody ever done, excuse my terminology, Indian burn? When they go this way and it just, it hurts, right? It's kind of a burn feeling. That's going to be our Ruffini's organs, that stretching is felt by those. Merkel's discs, those are there to register light touch. And the Meister's corpuscle, we have a Pacinian corpuscle up here for pressure and a Meister corpuscle for 2-point. We're going to try a 2-point distinction. What that means, if you take two little points, like say two toothpicks together and touch them, sometimes you can feel two things touching, but sometimes you can only feel one thing touching it, even though the two things are still touching. That's called a 2-point discrimination. And it is our Meister's corpuscles that are moved about, so in some places you can't feel it because there's no Meister's corpuscle associated with that area. But it's really, really close to the other one, right? That's 2-point. Then our proprioceptors, so our cutaneous, our proprioceptors include the muscle spindle and the Golgi tendon organ. Golgi tendon organ. These are found deep in the body, okay, in joints and in deep muscle. Okay, we have something called a spindle and something called a Golgi tendon organ. So I'm going to take you to our picture of skin, and we're going to see if we can find these items in there. Okay, so our thermoreceptor right here, since it's heat or cold, which one was that? Did y'all have that?
Smell. That wasn't the smell.
Which one was that? It didn't really tell us, did it? Okay, doesn't really tell us. Meister's corpuscle, that is, yeah, the 2-point touch. Nociceptors are down deep, that's for deep pain. Look, it's getting stuck with a needle up there. Here's a pinch. The Pacinian corpuscle, you know that it's got pressure. Look how low the Pacinian corpuscle is. It's deep down into the dermis. In fact, it's in the hypodermis, right here. All right, so next time we will look at, if we have time, I think I have another picture, some of the slides of this. Here's our Ruffini's, our Pacinian corpuscle, our Meister's corpuscle, our Merkel's disc. They're all found somewhere in the skin, in the dermis, dermis or hypodermis. And they're going to be feeling different things. Okay? Pressure on the skin. So we're looking here. The shape of the corpuscle gets bent when I put pressure on it, right here, causing sodium ion channels in the neuron membrane to open up. Sodium then diffuses, and the neuron and the concentration gradients change. Depolarizing, this is the whole sodium-potassium pump situation going on. We have a generator potential for an action potential. The greater the pressure, the more channels that open up. That would be frequency. If the threshold of the neuron is reached, an action potential develops and is transmitted saying somebody's pushing on me. There's pressure on me. You know, this might be something like, if you've ever had a massage? Mm-hmm. Have you ever had one of those like deep tissue massages that like hurt? I don't like those. I'm like, don't, you know, I want one of those like massages that feels good, you know. So that would be a deep pressure, but supposedly those deep tissue massages are really helpful to get your muscles all untangled and this kind of thing. And so they're good for you, and they release some of the chemicals, which afterwards you have to drink a whole lot of water, right, to make sure you're hydrated really well, that kind of thing. All right, we're out of the simple, we're going to the complex now. We're going to talk about smell. The olfactory anatomy. Detection is found in the olfactory epithelium in the superior nasal cavity, right at the top of the, what do you call that, the sinus cavity of your brain. There is that olfactory epithelium that's right there. I'll show you a picture in a minute. Each nostril has an olfactory bulb coming down to it. at the top there, from the brain where the olfactory neurons will synapse with association neurons. These are the only neurons in your body that regenerate. Remember we talked about neurons don't heal, right? Except olfactory neurons. So what that means is that every 2 months or so you have new olfactory neurons. So do you know, sometimes when people get really old, they can't see as well, they can't hear as well, but the sense of smell pretty much stays around. That's pretty cool, you know. We tend to keep our sense of smell. I did go to high school with a guy that was he was on the wrestling team, I guess. But anyway, he had been punched in the face so many times that he had broken his nose so many times that he had lost his sense of smell, which was really a bummer because the sense of smell makes our eating more enjoyable. If you can't smell it, you can't taste as much stuff. We'll get to that. All right, so here we can see in the nose, it comes up here. You take a big, if you're smelling someone goes, oh, smell this flower. You may go and take a deep breath and smell it, and you're going to pull all those little atoms up to the top of this, we call the cribriform plate right here. Here's the bone of the skull, and we're going to see this epithelium comes down here, and we have different glands and these are neurons that are reaching down here. Now, in order for something to be smelled, it has to dissolve into the olfactory epithelium. It's got some mucus layer here, so it has to be able to dissolve in there. All right, so the physiology of it. So the process, substance must be volatile and airborne. Okay, so for instance, if this table right here, we couldn't smell it, really, we can't smell it, right? But if I burned it, would you smell it then? It would be airborne and volatile at that point, okay? So therefore, we could smell it when it started. We'd say, oh, something plastic is burning, and we would know it was plastic right there. It has to be water and fat soluble to get through the mucus layer in order for us to smell it. The substance, it has to dissolve, and then there's a chemoreceptor that's triggered, and it triggers an action potential which sends it to the brain. It's a very low threshold. You don't need very many action potentials to smell something very well, okay? So I'm going to talk to you about propane. Do y'all have propane at your house? You know, they just told us that, gas stoves are like dangerous. I use a gas stove. I wouldn't want to use anything else. But it turns out the propane there's nothing in the propane that we can smell. We cannot smell propane, okay? So we don't have the receptor for it. Maybe there's some creature out there that can smell it, but we cannot. Now, it turns out that when there's a gas leak in your house, that could be very dangerous, yes? A little spark and their whole house is kaboom, yeah? So what we've done, okay, in fact, y'all, I don't know if you've heard this story. It happened in Texas. I wish I remember the name of that school. It was a school up in East Texas. I think it's East Texas. Yeah, I'm pretty sure it's East Texas. And a big school where the kids from all around all came to this one school because they were in the oil patch. So all their families were working in the oil business, okay? And these kids were in this school that it was pretty new, and down in the basement there was a gas leak, and they didn't know about it. And so down in the basement they had the shop class, and the shop teacher went to go turn on the little saw to start cutting something in this shop class. The spark from that, the whole school blew up. It killed something like 500 people. Maybe 300, but a lot of them being children who were in school. And interesting story, because some of the kids were away at a sports event, so they missed that day. Some of the kids were waiting outside in a bus because they were from like a elementary school that was coming to pick up the kids at this school. It happened in the afternoon. So when that happened, everybody said, oh my goodness, nobody had any clue that there was a gas leak. The gas was coming directly from the oil field straight into the school, and everybody in the Texas legislature at that point said, we've got to figure out a way to fix this. And so what they did was they passed a law. Now it's a federal, it's in all 50 states, so maybe just every state passed the law, which was natural gas companies, propane gas companies have to add something to the propane so we can smell it. So if when I told you can't smell it, you may have said, well, I can smell it. It's got that very distinctive, that's not the propane, that's the stuff they've added to it. And so like at my house, I've always noticed, I have one of those big, propane tanks like this, and I've noticed that if I can smell it, especially if I go into the room where my dryer is, and that's going to use the propane to heat the dryer, and if I walk in there and get that little hint of that smell, I know that my propane's low in my tank, and I'll go check it, and sure enough, it'll be low. That's because the stuff they put in there kind of sinks to the bottom, but it gets released even in higher concentrations when there's less gas in the thing, and so you can smell it. Okay, so we add that to it as a precaution, so that is now a law that's been passed. All right, let's take a look at that, those nerves that are coming in. So this is the olfactory bulb that's coming. Remember, we have two of them coming over the side here, reaching down to the cubiform plate. Right down here, we can see our little neuron and our little nerve endings right there. Here comes something that dissolves on it, and when it dissolves, and I have a receptor for it, I smell it, and I have a part in my brain that says, yes, I smell it, and there's also a part in my brain that remembers the smell. I smell popcorn, I know it's popcorn because I've smelled it before. Okay? So we have, remember, those are two separate places in the brain that are registering those. Okay, the gustatory sense is the sense of taste. The sensing organ is the tongue. It's covered with papillae. I'll show you a picture of papillae here in a second. If you've ever looked at your tongue, it's bumpy, right? I'll let you see a real close-up. It's kind of creepy, actually. About 10,000 taste buds. Interestingly, some of those taste buds don't have any taste ability. They just have a place for the chemical to rest for a minute so it can dissolve. The filiform papillae are numerous. They're the big ones back here, but they don't really taste. They just absorb or hold on to the liquid. The circumvallate papillae are the largest. I'm sorry, the circumvallate are back here. The filiform, where are those? I thought that said fungiform. Those are all over the tongue there. And a folate on the sides of the tongue. So I'm going to show you a close-up picture of this, and it looks like that. So these might be the filiform, and the fungiform are the red ones. Filiform and fungiform, like this. All right, so notice how there's a lot of spaces there. So I put something into my mouth, and if it dissolves, then that in my saliva of my mouth, the chemical reaction that takes place, it's going to be sitting down in this area, and then these guys right here have the receptors to taste it. Okay, not everything has a taste to us. For instance, water, sometimes you'll taste water, say there's no taste to this water, it's just water, no taste. But if you go to a new place, you say, oh, there's a taste in this water, because they have some sort of like mineral or something in there that we're not used to. We used to think that the tongue we could map it out, okay, that we would have sour taste were more evident on the sides of the tongue, salty and sweet, sweet right at the tip, salty just behind it, and bitter in the back. We've decided that is not the case, that everything can be tasted on all parts of the tongue, except maybe a little more sensitive on I've had students before like take a Q-tip and right in the middle they couldn't taste anything. So there might be something to it, but we don't teach it that way anymore as the mapping out the tongue. This is a close-up picture of what's happening inside your tongue. This is a taste bud, okay? You can see there, this right here, y'all, is a little gustatory hair. So your tongue is hairy. That's weird to think about, right? But it turns out hairs, we call them, are going to be found in lots and lots of the sensory organs because the hair can move. It's small enough it moves. And in this particular case, the chemical the dissolved chemical would end up in here, and that little hair is collecting that data. And if you look at it from the side, it looks like this. Now you can see why it's called a bud. All right, a taste bud. So here's the pore, which is what we're looking at in this picture. And now it's going to dissolve right on there, and these nerves right here are going to take that that stimulus and send it to the brain. Okay. Notice there's also a physical stimulus. You know, if you touch your tongue, you know it. You can feel the touch of it, right? But the chemical part is the stuff that's dissolving. If you have something on your finger, sugar or something, you can taste it.
So with the tongue, what's it like when you burn your tongue and you can't taste anything?
Oh yeah, so you've just desensitized those for a bit. They have to heal.
It comes back a few
Yeah,
days later.
they have to heal. They heal pretty quickly, just like most mucous membranes will heal quickly, because a lot of blood supply there. All right, what are we at? Oh, the ear for balance and hearing. So this is part B. All right, the outer ear is where sound is collected from vibrating air. Okay, so this outer ear is called the pinna. If I cut off this thing, could I still hear? Yeah, that's the hearing doesn't take place here. And also, if you cup your hand over your ear like this, it extends how big that is, and you can hear me better. You just catch, it's like a little funnel. It's just catching more sound as it's coming in through there. It's coming through vibrating air, and then it goes in through the ear canal, down to the ear drum, into the middle ear, where the air continues to vibrate, and we have these really small bones I'll show you in a minute. And we have the inner ear is fluid filled, where the vibrations will go through the system even further. So it's all about vibrations traveling through air, and then the vibrations travel through liquid in the inner ear. Anybody here have ear problems when you were little? Ear infections and you had to get tubes put in your ear?
I didn't
Nobody?
get tubes. My grandma got like newspaper and like rolled it up, stuck it in my ear, and lit it on fire.
And burned it? Yeah, that's supposed to help pull out some of the wax and stuff like that. Did you have infections or just kind of
I
gunky
had
ears?
that, and then I had to go to the doctor, and they stuck something in my ear, and I just remember like opening my mouth and my ear would pop.
Okay, yeah. So, and was it painful?
Yeah.
Yeah. So I'll show you. I don't think they do these ear tubes very much anymore. When my daughter who's 35, she had it when she was little, and I think they've moved away from it. I think they're trying to figure out how to deal with it without putting. Have you heard of ear tubes though? Yeah. No. Okay. All right, we have this. I'm going to show you the anatomy now. It's pretty cool stuff. All right, so we have the auricle or the pinna, or the ear leather. Sometimes they call it, and animals will call it the ear leather. It's going to come through the ear canal, so the sound's going to come through, and it's going to vibrate. We know the eardrum will start vibrating. Then it'll vibrate these 3 smallest bones in your body. And this is air-filled. This is called the eustachian tube. So that middle ear connects to your throat. So, you know, if you're on a plane and your ears go ring, and then you chew gum or swallow or you do this, it opens it up. That's the eustachian tube. It's just regulating the pressure. So the reason why your ears act funny when you go on a plane is because you have a change in pressure. Same thing if you go up a mountain. Yes?
Is it bad to use Q-tips in your ears?
And it does
Okay.
because it pushes back.
What happens, so here's the deal. We have wax in our ear, yes? It's there because God designed it that way. So wax is good. Sometimes we get an overproduction, it feels, if you stick a Q-tip in there, you're going to end up maybe pushing the wax a little farther in. I do know a kid one time who had a ear swab, and he was really digging in there, and he broke his eardrum, and it was bleeding and everything. Now the eardrum healed; it was okay, but so there can be a problem. So if you're careful, I will admit and confess to you, I do put an ear swab in my ear because my ears itch. So yeah, I got to get that water out of there from my shower or whatever. All right, so we have air vibrating the drum, we have air vibrating these bones, and then once it gets into this right here, the inner ear, this thing right here is called the cochlea, which literally means snail. Looks like a snail shell. It's made of bone, and it's hollow, and it's got liquid. So there's liquid inside here and liquid inside these called the semicircular canals, which we'll get to here in just a second. All right, once again, the eustachian tube is used to equalize the pressure because that's air there and air there. All right, so let's talk about balance first. Okay, I'm going to go back for just a second before we write this down. This area right here, this is called semicircular canals, right up and right about here, called the vestibule, is where balance is achieved. Okay, think about it for a second. This is fluid filled. All right, if it's fluid filled and I start it moving, what's going to happen with that fluid when I stop moving?
It's gonna slosh around.
It's going to keep going, right? It's going to have some inertia to it. That's what causes dizziness. All right, so we're going to see how this part of our ear is going to help us with balance and, of course, dizziness. Okay, so the vestibule is that part that's just outside of the semicircular canals. We're looking at these semicircular canals here. There's 3 of them, but we're only seeing 1 of them right there. This pink stuff is supposed to be liquid. Okay, so it controls static equilibrium. In other words, y'all are sitting still right now, and your body knows it because the fluid inside your ear is reasonably stable. It's not moving around too much. There's something called the saccule, which you see here. It's a little patch of nervous tissue. And the utricle, right next to it, another patch of epithelial tissue with neurons coming to it. The macula of the utricle is parallel to the base of the skull. I think I'll show you a picture of this in a second. And the macula of the saccule is perpendicular. Therefore, your ears know if your head is going like this, or like this, or forward, or up. It's like a little level, right? A little level inside there. That as the fluid passes by it, your body knows it and allows you, instead of, when I do this, if I didn't have that capability, I would just keep going, right? And just fall over. And so dizziness can affect that. So because my macula and saccule will be confused by all that motion in there, and it'll cause you to walk kind of crooked. And drugs and alcohol can do that to you as well. All right, so we can see here what's happening. It's so cool. The semicircular canals are coming off of that, right? right, so the fluid is going past the utricle and the saccule. You can't actually see it, it's right down here. And then we're going to see dynamic equilibrium. Okay, so static equilibrium is when you're sitting still. Dynamic is when you're moving around. Okay, running, playing, whatever you're doing. And what happens is that these semicircular canals will know, because there's one going up to the side and straight out, so X, Y, and Z. So therefore, as I'm moving, let's say I'm doing a cartwheel, couldn't do that if you paid me, but like this, it's going to know that I'm upside down and coming back up, and it'll go into those semicircular canals and know that there's a gelatin-like cupula. You can see it right here at the bottom, right here, that slides over something called the crista ampullaris. There's literally these little otoliths, which means ear stone. So you do have rocks in your head. You move your head to the side, you fortunately we can't hear it because it might drive us crazy. But they're kind of rolling past this little called the cupula right here, rolling past there. Mechanoreceptors, that's motion, sense the motion and send an action potential to the cerebellum. Remember, cerebellum is for movement, coordination of movement, right? So your ears are going to send a lot of information to your cerebellum to coordinate your movement. So you can do, if you're doing something real, you're swinging a bat in baseball, you got a lot of movement that's going here like that, your head's moving around, it's going to know which way you're going. All right, hearing is the star here when we talk about the ears, right? Once the sound vibrations are collected in the auricle, that's the other part here, the tympanic membrane, which is another word for eardrum, begins to vibrate, attached to the bones. We'll see these called the malleus, the incus, and the stapes, otherwise known as the hammer, the anvil, and the stirrup. The vibrations travel through the air-filled space until the stapes, the stirrup, vibrates the oval window on the vestibule of that fluid-filled space. Here the vibrations, it passes to the fluid-filled inner ear. The vibration passes through the cochlea. That's where hearing takes place. So in the inner ear, the semicircular canal and vestibule are responsible for balance. The cochlea is responsible for hearing. All right, so there are two different parts there, and that's why we put balance in as a sense. All right, the cochlea, where they're the mechanoreceptors, remember motion, sense the sound waves if they move over little hairs inside the cochlea. Once again, sensory hairs are going to be moved by this fluid. They send action potential to the brain to interpret it, and as we can hear it. So once we can hear it, we know there's sound, we know there's pitch, this kind of thing. We'll also have memory. I know that's the sound of a trumpet. I know that's the sound of my mother's voice. I know that's the sound of a song. I know I'm going to sing along with it. Remember, that's two different parts of the brain that'll remember those two different things. At different points along the cochlea, we have different frequencies. All right, so I'm going to show you a picture of that here to help you understand a little bit better. So different parts of that snail thing pick up high pitches versus low pitches, and that has to do with the frequency of the sound. All right, so here's the drum, eardrum, and here are the three small bones, the hammer, the anvil, and the stirrup. That foot plate right there, see how it's kind of oval shaped, is going to be placed up against what's known as the oval window. All right, so the real name of these bones are the malleus, the incus, and the stapes. It does look like a stirrup. And that little end right there is that oval part that's going to touch the oval window. And I'm going to show you, give you an idea how small they are. So they're really tiny. They are the smallest bones in your body. And then I have one next to a dime here to show you how. And also I like the way you can see how well. And I think hammer, anvil, stapes, stirrup is a pretty good description of them. Because this one's hitting this one like a hammer. The anvil's vibrating and vibrating that. So vibration, vibration, vibration. Okay, so take a look at it. We can see the air vibrating the drum, which vibrates these three little bones. And then it's going to vibrate the oval window, which will go into the vestibule for balance, the semicircular canals, so this is static, dynamic equilibrium, and also sending information right through there. So it's sending two different types of information through. Okay, so I think I have another picture of this, so you have to bear with me while that's still moving right here. So if you get an ear infection, so my daughter who had many ear infections, what happened was this area that should be air-filled became fluid-filled. Because of a cold, a lot of, mucus, stuff like this. So this became filled with, and it got infected. And because of that, she had to be on antibiotics to kill, because it was painful, right? So finally, after several bouts of that, what we did is we went in and put a little tube right in there, looked a little bit like a spool, and that allowed the fluid that came in this area to just leak out of her ear. Therefore, it came out of her ear. It's kind of gross, but it allowed that she didn't get infected back in there, right? And the only problem was is that when she took a bath or whatever, she's a little baby, right? She's 18 months old. We had to put these earplugs in there so no water got down, because the water would go through that little tube and get into her inner ear, and that would, middle ear, and that would be a problem. I had a student many, many years ago, Sam, his name was Samuel, in Austin, and he told me one week, he said, Mrs. Gasper, I'm not going to be here next week because I have to have surgery. I said, oh, I'm so sorry. What's going on? He goes, well, here's what happened. When he was younger, he had multiple ear infections, and at one point in one of his ears, the infection dissolved those 3 bones. So he had no bones in one of his ears. So he was completely deaf in one of his ears. And I was like, oh man, what are they going to do? He said they're going to put in some artificial bones. 2 weeks later, he came back, he could hear in that ear, because they had done something to somehow connect that to that and have movability. Pretty cool stuff, yeah. I told him I'd be telling that story to everybody. He said, that's cool, we can do that. Okay, now once the vibrations make it into the cochlea, we know what happens when it gets to the vestibule and semicircular canal. We have that whole thing with your otoliths and stuff. But now let's see what happens when it gets into the cochlea. All right, so you can see here what it is. It's actually 2 sets of tubes, yeah? And I can't remember which, it starts at one place and ends at another one. So the vibrations come through the liquid in this part, come up all the way around, come around here, they turn around and come all the way back, okay? So when they're in here, they're going to hear 20,000 hertz, and then 7, 5, 4, 3, 2, 1500, and then down here we're going to see 1,000, 800 hertz, 600. These are all, the hertz are the frequencies of sound, okay? And then when we get down here, 200 hertz, okay? And so but it does go all the way back to send that vibration out. So what's happening? High frequency waves are picked up 1,500 to 20,000, so from here to here in this part, and then back on this part we're getting the lower frequency. Medium 600 to 1,500, so here to there, and then the other one's 400 to 200, right there. Okay, so it's just the way they're accepting those, so we can hear the different sounds. Let's see here. This is a little bit frenetic here. Oh, look, it goes from the top to the bottom. So it goes to the top first, around and back here, and wherever it hits those little hairs, we will get the change in the frequency. Before I move on, there are people who are deaf. Do you know anybody who's deaf? Okay, and it can be caused by various things. Like this kid, he had that disease that caused that, and it was able to be cured. But usually what happens, there's a defect of some sort in the cochlea, naturally, right? He had a defect in his middle ear, but the defect in the cochlea, it mostly has to do with that fluid drying up and those little hairs not being sensitive to fluid moving past it. So there is something called a cochlear implant. Have you heard of this? A cochlear implant, where they put a little computer in there, and you have a little magnet on the outside of your head right here, and it can bring in those vibrations and interpret them. It's not nearly as good as real hearing, but it does give you that hearing back. I don't know if y'all know who Rush Limbaugh was. He was a radio guy. He made his living on the radio. That means talking. And in order to talk, we have to hear, yes? Have you ever, if you met a deaf person and they speak, their speaking sounds just a little off, right? Because they can't hear themselves. Okay, so our hearing affects our speech quite a bit. And so here's this guy who speaks for a living, and he went completely deaf. And I was a big fan, and so I would listen to him, and I noticed his voice changing. And later he came back and said, well, and then he had got a cochlear implant, and everything was great. They only did one ear, because the hope was they would find something to fix the other ear. But they never did, so much later in his life he got a second cochlear implant. So he was able to hear that way. All right, vision. Here we go. The eyes are specialized organs tasked with complicated job of requiring an exquisite cascade of biochemical events. Yikes. Okay, all these things have to happen in order just for you to see something. They're delicate organs. They have to be protected by these bones in your skull, right? Skull bones hold them securely but gently at the same time. Facial muscles hold them in place while allowing intricate movement. So we have very, very intricate musculature around your eyes, because think about it, your eyes can move in pretty good and pretty quickly. They can move around pretty quickly, right? So not only do we have the muscles moving your eyes back and forth, up and down and around, this kind of stuff, we also have the muscle in your iris, right? It closes and opens to let the amount of light come in. That's also a muscle that's doing that. It's a circular muscle inside your eyeball. All right, so these are called the orbits in your skull, and it's hard for an eyeball to be knocked out of its orbit. That's very difficult. Can it happen? Yeah. I will tell you, at the vet clinic though, there's a certain kind of a dog that has a pushed-in face, you know, like a pug, and what's that one? Bulldog. Bulldog is, yeah, they do, but not because their chin comes out so much it kind of protects their eyes. There's another one with a really flat face.
Chihuahuas.
The pug definitely has a flat
Chihuahua
face.
has big eyes.
This one I'm thinking is kind of a furry animal, like a lot of furry. Anyway, I just remember one of those critters came in one time and its eyeball was hanging out of its head. Yeah, it had been attacked by a dog and the eyeball was fine.
But it was sticking back in there.
Pretty much.
So could it like see still?
I think it probably could see, but it couldn't
So
move
it's
around.
like two different like.
It wasn't like hanging like this going, because the muscles are up here to do that. It was just hanging there like that. And the dog's just going, it's just hanging. And we were like, we did, all of us kind of went, oh, you know, so we had to go put him under anesthesia. And then it could, but our bone is so protective. I mean, because you can't imagine how big your eyeball is. You think, well, it's this big. No, it's huge. And it's really stuck in there. Yeah. So to get it out, maybe if your bones were broken, it could come out. This little dog's bones weren't broken or anything because his face was so flat. He didn't have as much bone holding onto his eye. So we had to sew the eye shut for a little while, but that's kind of normal in dogs. When we have eye trouble, we'll sew their eyes shut to let it heal. Then we took the stitches out and the dog's eye was fine.
You know how in the Old Testament they used to like gouge people's eyes out? That
It's-
must have been a dirty job.
Well, and I will say it's kind of easy to do that because you can just pop the eye. Sorry. It's full of fluid. It's full of fluid.
Oh, it is?
Yeah.
For like they use a melon molar to get their eyes.
Oh, that'll do it. Okay, so let's take a look at it. Here's our
I'm
eye.
scared now.
right
My
here.
glasses.
I will tell you this, something interesting, y'all, that I just learned about eye color. You know, everybody's always fascinated with eye color as far as genetics go. It turns out, did I already tell you this? There's only one color of eye, and it's brown. That's it. That's the only color there is. That's on the back part of your iris. Yeah.
Yeah.
And so what happens is, if you don't have a lot of melanin, that gives you the dark color in your skin and your hair and this kind of stuff, the outer part of the iris, this part right here, doesn't have much melanin in it. And so when my eyes are green, it's really just the light bouncing off my eyes. Blue eyes, just the light bouncing off of it. Okay? It just means you have less melanin in the front part of your iris. Isn't that weird? Just like the sky is blue because light is refracting through it, my eyes are green because light is refracting through it. That's why people with lighter color eyes, you're like, oh, my eyes look bluer today. My eyes look greener today, that kind of thing. It's because it's refraction of light. That's all it is. Pretty cool.
Well, is it true that people everybody's born with blue eyes like Cass?
Yeah, that's true, and it has to do just with the development as it's going. You know, have you ever seen a newborn babies? They're kind of that bluish, but they're real opaque looking.
Mm-hmm.
Yeah, remember they're not processing a lot of visual information quite yet, but as they move along, that melanin will settle into it. Okay, settle into it. All right, so let's talk about the anatomy of the eye. It's pretty amazing. Okay, first of all, it's round. We have two of them. That gives us the ability to see things. We're going to talk about this next time. We'll check for your blind spot next time. Somebody like, you know, who's lost an eye for some reason in war or whatever, you know, then their depth perception will not be as precise. Okay, so, you know, we need our depth perception. Like if we're going to shoot a gun, we need to have a good idea of where that target is that I'm shooting. All right, first of all, it's made up of tough tunics. So I told you, okay, you can pop the eyeball, but it's not easy. You know, you put your eye out, kid, right? That kind of thing, right? You can pop the eye, but the tunics are very, very tough. I mean, you can even, like, if you push, you can feel, that's just really tough. Of course, they have a lot of nerve endings, so it hurts when you do that, so be careful. There's the fibrous tunic, the vascular tunic, and the retina. So the retina is the innermost tunic. The retina is the vision spot. That's where vision takes place. It has in it some specialized cells that have photoreceptors on them. Okay, that's where the photoreceptors come in, right in here, in the retina. Outside of that, we have our vascular tunic. What does vascular mean?
Blood
A
flow.
lot of blood supply. Okay, so we have all that blood supply like this in your eye. So think about this, y'all. When I told you there's only one color of eye and it's brown, okay? You ever heard of a person or a bunny or something with red eyes? That's just the blood. That's just the blood we can see. That's all it is. It's the reflection and refraction of the blood in the tunic of the vascular tunic right
Is
there.
that when you take pictures, your eye?
Exactly, because actually the light goes straight through your pupil. It bounces off the retina, which is highly vascularized, and you see that red kind of glare. It's the blood that you're seeing, the reflection of the blood. That's why the no red eye feature actually flashes a light at you a couple times, then it takes the picture. You know this? It's also technology that's kind of gone by the wayside, because I think our digital cameras are better at taking pictures in lower light.
My grandpa has something with his eye where he goes and gets a needle put in it and it has to get a shot in it or something.
Is it glaucoma?
I
Does he have
think
glaucoma?
so. And then he's got like if he looks, there's like blood like coming out the side of it.
So what glaucoma is when there's excess of fluid inside the eyeball, and it's painful. It's like pressure from inside it, and the eyeball itself is swollen, and so they go in there and they relieve the pressure by taking fluid out.
That blood is in there. He says it feels like there's sand in there. He can't get out.
Oh,
He
how
can't
awful!
sleep and-
Sometimes they'll leave a little tube coming out of the eye, and it just drips all the time, like that. For people with glaucoma, fortunately, it's not super common. The other tunic would be the fibrous tunic. That's the white part. Something that's super cool, y'all. What we call the white of the eye is made of this fibrous tunic. Okay, when the white of the eye gets to this part right here, we have something called the cornea. Okay, so we call the white part the sclera, all right, and it's made of this fibrous stuff. And then we have the cornea that we can see through. It's the same tissue, but it's opaque white here and it's clear right there. That's weird. Can you imagine if someone didn't have a clear cornea, it'd be white all the way? That's a scary movie right there. When they open their eyes, it's all white. That's scary, right? That's a scary movie. Yeah, very creepy. So that's why we have to have our cornea has to be clear, because light has to come through it. I just think that's really fascinating. It's not a whole separate thing. It's part of that tunic. That's how tough that tunic is. It just gets clear right there. All right, so the tough tunics. Now let's talk about vision. Here's the eyeball. And we're going to take a look. This is a closer look at the retina, or retina, if you want to call it that for spelling purposes. And we're going to see, look at all the action that's taking place there. So light comes into this tissue, and these little cells gather that information, and these cells start to process it. All right, so we have different kinds of, these are neurons. These are photoreceptor neurons. And then these right here are fancy little, also photoreceptors, some called cones and some called rods. We have a lot more rods than cones. Retina is highly vascular, lots of blood supply. The photoreceptor cells are deeply embedded into it right down in here, past the little tunic. We're going to pull that off the eyeball next week. The rods are going to take black and white. Okay, so we're going to get into the idea of light and color. Sometimes students get a little freaked out about this because color is a function of light. So the fact that the lights are off in this room, that means there's less color in this room. It doesn't mean the colors change or anything's different about it. It's just not as much light bouncing off of it. When I turn the light on, the colors will be brighter because there's more light bouncing off of it. Okay? So think about it. You're out on a summer evening and it's a beautiful day, and then the sun starts going down. It gets dimmer and dimmer. All of a sudden, think about it. Everything looks kind of shades of gray, right? The colors are less pronounced. And it's because there's less light. So I like to tell my story about my house. We were painting my new house, and I wanted it to be gray and white. And my husband picked out a paint, and he puts it up on there, and we're looking at it, but it was evening. And I said, yeah, that's great, I like that, you know. The next day in the bright sunshine, it's actually a pale blue. But I didn't know that. It's called French silver. I thought that sounded cool, but my house is really kind of a pale blue and white. So, I don't know.
Do you
So
still
look
like
pale
it?
blue and white. I do like it. It looks more pale blue than gray, yeah. We've since repainted, so maybe we use gray the next
Just
time.
invite people over in the evening.
Say again?
Just invite people over in the evening.
Yeah, that's true. And say, look at my gray and white house. Yeah. All right, so the rods are responsible for collecting the data that's considered black and white. Now, when I have bright light shining, my cones will pick up the color. Okay, so the color, there are 3 colors that we see, and those 3 colors are the primary colors of light, and we mix them all together. There is some data out there to suggest that there's possibility of some people having an extra cone, an extra something to see another color. I don't know. But apparently some people see things more vibrantly than other people do. But I don't know how you could prove it, because I can't go in your brain and look through your eyes. They talk about dogs being colorblind, right? Well, how do we know that? Yes?
Isn't it dogs like red colorblind, like they can't see red colors very
They
well?
can't differentiate. So colorblindness isn't that you can't see any color. It's you can't differentiate between colors. Between red and green, you can't tell the difference between them. All right? So I think I'll show you some examples of that next week and make sure you're not colorblind. If it's going to be the guys, if anybody, because it's an excellent gene, so it actually shows up in men versus women. Not that women can't be colorblind, but it's much more common in men. Okay, so the process by which all this takes place just to be able to see something, we're just talking about receiving light and making vision out of it. We're not even talking about focus and all that stuff. The cascade of biochemical events here that takes place have led people to get saved. When they study it, they say this is absolutely, utterly impossible unless every bit of it was invented at the same time. There's no way it could have evolved through millions of years. It can't have done that. And even Darwin said to suppose that the eye could have been formed by natural selection over millions of years seems, I freely confess, absurd to the highest possible degree. Didn't help him believe in God, but it did help lots of people become Christians because of it. So let's take a look. There's our photoreceptors. And here is our retinal, it said retinal pigment epithelium. You're going to see next week that the retina does have a color, and it's kind of this pretty opalescent black color. It's going to be able to absorb all that light that comes in there because of the color. Let's see. It says here the retinal pigment epithelium further complicates the complexity of the eye screening design. It's like fiber optic cables funneling the proper wavelengths of light into the right place. Okay, here's our blood vessels down here. Okay, and all these photoreceptors pulling it through there and amplifying it essentially. All right, so let's take a look at it. It's round, first of all, right? The pupil is a hole. There's a hole in your eye. So it's that black hole, and that's so light can come into it. We have this thing here called a lens. All right, so light comes through here, and the lens will bend it. So the lens is responsible for refracting the light that comes in. And depending on how good your lens is, y'all are young, and because of it, your lens is nice and pliable. I'm old, and my pliability has decreased. My lens is kind of stagnant. So I never wore glasses in my life till I got old. And these are called, cheaters or readers that help to. This is another lens. It's going to help. It's for me. I can't read things up close unless I have these on. Because it's because my lens won't change. It won't stretch out like it used to. Y'all's lenses are stretching out. Okay. And then we're going to see, look at all that blood vessel here. Notice right here we have something called the optic disc. This is the optic nerve coming into the back of the eye in that little spot right there. In that little spot where the nerve is coming in, you cannot see anything. It's called your blind spot. But because we have 2 eyes, it compensates for it. You'll see next time, we're going to cover eye, you're going to get to a place where there's a place, when I hold something up, you're not going to be able to see it because it's in your blind spot. Also, the optic nerve, if you remember this from our previous module, the optic nerve is strictly sensory. So technically, if I touch the optic nerve, you can't feel it. It doesn't have any pain on it. It doesn't have any of the somatic part of it. It only has the sensory part of it. And it's pretty big. I think it's one of the largest nerves, certainly the largest sensory nerve in our body, right there. All right, anything else here? There is a spot. It's kind of like the light coming in and shining a little projected image on the back of your eyeball, okay, like a movie. But because of the way light travels, what do we know about the image? Because of the properties of light, travels in straight lines, the image actually gets projected upside down on the back of your eye. Same thing happens if you look through a little pinhole camera or something like that. The old-fashioned cameras, the people would be upside down. Yeah, okay. And so let's bring it in a little closer. You can see right here the light coming, straight lines, bending through the lens, focusing right back here on the fovea, passing through the vitreous humor and the aqueous humor of the liquid of the eye right there. And that also acts as a lens. Bending the light. Keep in mind, light is also coming straight through, like this. The middle of the flower is visible right there. Okay, so people would say, why is this happening? The answer is because light travels in straight lines. Now, sometimes our eye might not be perfectly shaped in order to receive the data at the right spot on the retina. And so because of that, we'll have a misshaped eye right here. So it's not focusing in the right place. Okay, it's focusing up here instead of right here. Here it's focusing too far behind. So we call this nearsighted and farsightedness. So some people whose eyes, like for instance, so Zoe, you wear glasses. So what are you?
I can't see far.
You can't see, you're nearsighted. So you can see near, so you're called nearsighted. So at a distance, things look blurry. So for driving, you need your glasses.
Not legally.
Not
Because I'm
legally,
not
okay.
that blind.
Do you need them to see this screen?
Not if I know.
No? Okay. Yeah. So my vision problem is called presbyopia, just meaning things aren't focusing right, which would be farsighted. I can see far just fine. Near, it gets blurry, so I need to look at it. So farsighted means you need glasses for close up. Nearsighted means you need glasses for depth vision or far vision. And so it has to do a lot of times with the lens shape or even placement for that matter. And so we end up with a blurry section instead of a in focus. Let's go to here we go. Our myopia, nearsightedness is the top one, yes? No, right here. And it's focusing too soon. Hyperopia, farsightedness, it's focusing too late against the retina. Astigmatism is there's multiple things that you're trying to, oh, that's what it is? Okay, it's trying to focus in multiple locations, and what you're trying to do is just try to get those guys into agreement. So the lenses will change where the light, how the light's going to bend. That's why you need a doctor to figure out what lens. He puts different lenses in front of you, and you say, oh yeah, that's in focus. That's okay. That's not in focus. That's in focus, kind of thing. Yes.
Is it like both your eye? Normally, both your eyes have like the same like bad vision, or is it?
Not
like,
necessarily. Oftentimes it can be different for each
My
eye.
right eye's way worse than that.
Oh yeah, and so your lenses will be different that way. Your lenses and your glasses will be different that way.
Yeah, don't people have like one dominant eye and one?
Yeah. Well, but this is not about dominance. This is about the shape. So therefore.
Well, like they're better on one than the.
Yes, but that doesn't necessarily have to do with focus. That just has to do with, like we're right-handed or left-handed, or right-eyed or left-eyed, right? That's just however that's chosen. Left-handedness and right-handedness are not genetic. We don't know what causes it. But left-eyed, right-eyed, so some of you could be right-handed and left-eyed. You know, some has like left-eared. You hold the phone down to your left side instead of your right side. You know, one way you're more comfortable, right? So it's different for all of us. Presbyopia, that's me. That's when the lens just gets hardened and it causes the focal point to reach past there. So I need some lenses to correct it so that it comes and focuses and has the little movie projection, if you will, in the correct spot on the eyeball right there. All right, so we can correct it with lenses, either using eyeglasses like this. Who does contacts? Anybody have contacts?
I do both.
Have you done contacts? I just don't know if I could stick my finger in my eye.
It looks different than from wearing glasses. 'Cause
Oh, really? Okay.
I just don't like. That's why I wear glasses for. Because.
You prefer your glasses?
Yeah.
Okay. I just can't imagine I'd be going, oh, when I'm trying to stick something in my eye. My eye would be like, no, don't do that. So that's way to correct it. All we're doing is just changing the way the light goes through the eye. That's all we're doing. We're helping the lens to refract the light in such a way that it focuses in the right spot on the retina back in the back. All right, and it looks like we're done. I could have talked about eye diseases. Usually I don't make it through the whole lesson. I'm not sure. All right, so next week we'll have a quiz over this, and some people consider Module 9 lab their favorite lab. So we do a bunch of fun stuff. Remember to bring your study guide.